Hi students, this is Mrs. Foy, and this is a lecture on the second half of photosynthesis, the synthesis part of photosynthesis. And we're going to start off talking about the Calvin cycle. So if you're watching this, we've already talked about the light-dependent reaction, and now we're talking about the Calvin cycle and what is the plant going to do with the ATP and the NADPH that it has created from the light reaction. And the answer to that question is it's going to make some sugar, basically out of thin air. It's going to take carbon dioxide from the atmosphere and it's going to fix carbon dioxide to a series of organic molecules, and we're going to end up making some sugar. So that's what we're going to see. And of course, this is happening in the chloroplast. Specifically, it's happening in the stroma of the chloroplast. So what's going to happen is you're going to see a cycle with a bunch of different organic molecules, and it's going to look very similar to the citric acid cycle that is in respiration. These cycles regenerate the starting material after the molecule enters, so it's always regenerating the molecules it's going to use again. It doesn't just use it once and be done. It takes energy and invests it so it can use it again. And we are going to take the ATP energy. Remember, this is an anabolic process. This is going to require energy, and it's going to require some reducing power, some electrons. Remember, the cheat is to follow the hydrogens, right? That's going to come from the electrons that initially jumped off the chlorophyll and was replaced by the water and ends up on this NADP, and it creates NADPH. So if you are one of my students, then you are not required to memorize this whole cycle, so just take a deep breath, but you are going to have to be able to recognize where the carbons go, and there's a couple of molecules I'm going to highlight that you do need to know. So the first stage that happens in the Calvin cycle, which was named after a guy named Calvin who discovered it. Remember, this is also called the light independent cycle, and also the old-fashioned name is the dark reaction because it does not require light. So what's going to happen is carbon dioxide is going to come in. It's going to enter through the stomata of the plants, and the first thing that's going to happen, this first phase, is called carbon fixation. So the Calvin cycle has three parts to it. It's going to be the carbon fixation, then it's going to have a reduction cycle, and then the last phase is called the regeneration of RUBP. So let's start first. We have a carbon dioxide that enters, and the first thing that happens is there is a 5-carbon, see, 1, 2, 3, 4, 5 carbon molecule called ribulose bisphosphate, or RUBP for short. And there is an enzyme that catalyzes this reaction, and the enzyme is called rubisco, or ribulose bisphosphate carboxylase. How's that for a mouthful? And we call that rubisco, and that is the enzyme that is going to catalyze the carbon fixation cycle, where CO2 is going to be fixed to the 5-carbon starter molecule. An interesting fact, rubisco is one of the most abundant proteins on Earth. So that just shows you how important it is. So the first thing that happens is that there is this 6-carbon molecule ends up splitting into a 3-carbon molecule called 3-phosphoglycerate. Remember, this 3C is the nickname of this version of the Calvin cycle. So we're going to see that making sugar, there's more than one way to make sugar if you're a plant, and so this is called 3C photosynthesis. This is just the standard photosynthesis. So we'll go on from here, but I just wanted you to see that's a 3-carbon there. So the second phase, so now we have fixed carbon. We've literally glued a CO2 onto our ribulose bisphosphate. We make a 6-carbon molecule. It's unstable. It breaks into 3 carbon molecules called 3-phosphoglycerate. So the next phase that's going to happen is we're going to invest some ATPs and we're going to reduce. We're going to add some electrons to this molecule, and this is called the reduction phase. So we're going to spend 6 ATPs that we made in the light reaction, and we're going to use our NADPH. Remember, that was shuttling the electrons to the Calvin cycle, and this is called the reduction phase. And so there's a bunch of different intermediate steps that happens here. Every one of these is an enzyme, but it makes a molecule called glyceraldehyde 3-phosphate. And that glyceraldehyde 3-phosphate, about, if you think of them as 6 of these, you know that this cycle has made 6 of these. 1 in 6 goes on to be the starting material to make glucose. So technically, glyceraldehyde 3-phosphate is the product, and that goes on to make glucose as well as other organic molecules like proteins, like starches, but this is where the plant is actually making its food. But notice that 5 out of the 6 glyceraldehyde 3-phosphates are reinvested into making more ribulose bisphosphate. This also requires some ATPs to do this. So this is an anabolic process. And in this way, the plant is investing in the regeneration of the starting materials for this cycle. So just to keep some bookkeeping, you can think of this as per molecule, or more practically per mole, right? Because we're not just making one molecule at a time, we're making a whole batch, like a whole mole. So to make a whole mole of glyceraldehyde 3-phosphate, which remember is essentially the starting material to make glucose, you need 3 molecules or moles of carbon dioxide, 9 moles of ATP, and 6 moles of NADPH. One of these out of the 6 that are made, 5 of them are used to regenerate RUBP, and one exits the cycle to make glucose. So this version of the synthesis part of photosynthesis we call C3. And most plants, the great majority of plants on Earth, use this type of photosynthesis. And we call these plants C3 plants. So, for example, wheat, soybeans, rice, all the trees, or most all the trees, so about 95% of plants do this type of Calvin cycle, and we call those C3 plants because of this 3-carbon molecule that's generated here when CO2 is fixed. But as it turns out, in hot, drier environments, there are different types of photosynthesis. There is a type of photosynthesis called C4, and there is a type of photosynthesis called CAM. So C4 plants would be like grasses, corn, sugarcane, and we're going to see that they are going to make sugar in a slightly different way, which I'll explain in a minute. And CAM plants are yet another type of photosynthesis that's going to be done by succulents, which are desert plants, and plants like agave, which is a kind of a sweet plant, and pineapple. And the reason why this happens is a little bit more complicated. We're going to go through that. This is the most energy efficient, but it has a problem with this C3, and that problem has to do with getting enough CO2 without losing enough water. So here's the problem. It has to do with getting in carbon dioxide. So remember that the plant has to pull in carbon dioxide from the atmosphere, and in order to do that, it pulls it through its plant nostrils, or the stomata. But every time that happens, the plant also is going to lose some water vapor. And that process can dry the plant out. So it's kind of scary because the plant has to open the stomata to let the CO2 in, but then it also is risking dehydration by letting too much water out. And so there is going to be a different strategy to be able to open the stomata to get CO2 in, but in hotter environments, being able to not lose too much water. So this process of water being evaporated from the stomata is called evapotranspiration. And evapotranspiration is huge for plants. It's basically like plant sweating, but it has to do with the water cycle of the earth. It drives weather. It's very, very important. And it also helps water to be able to move through the vascular tissue that is going to deposit it up in the leaves where it needs it to be. Notice that oxygen, remember, is a waste product that was generated back in the light reaction and or the light dependent reaction. And so oxygen has to be released there. And you might say, well, wait a minute, I thought oxygen was needed for cellular respiration, which the plant does. You're right. But too much oxygen around can actually be bad for the plant, as we'll see. So one of the things that happens is, as we said, this transpiration process. So when you allow the stomata to open, then you will see the water moves up through the cells in the roots, and it basically is released through these little openings, the stomata, and that is a good thing. But if you leave the stomata open too much to let CO2 in or to let oxygen escape, the plant can sweat too much, evapotranspirate too much, and it can dry out. This is interesting. 1 acre of corn crops will generate 4,000 gallons of into the atmosphere a day. Okay, so evapotranspiration moves a lot of water into the atmosphere. So here's the problem. You open up your stomata, you could have too much evapotranspiration, you could actually dehydrate. So you might say, okay, plant, just close your stomata so you can conserve water. But the problem is that when you close your stomata, yes, you don't dry out, but then you can't get CO2 and you cause oxygen to build up in the plant because it's a waste product. And those conditions seem to favor a wasteful process called photorespiration. So photorespiration is a bad thing that happens when a lot of oxygen is present. So let me give you just a quick little history lesson of the Earth so you can understand why this is happening. So you have to go way, way back, right? So our Earth is, 4.6 billion years ago, and there was no photosynthesis that was occurring in the first 1.5 billion years. But then photosynthesis evolved, the process of photosynthesis, which is great because then organisms could basically make their own food. But there was no oxygen in the atmosphere, or very, very little oxygen in the atmosphere in early Earth. And notice this is the level of oxygen. Notice that it took several billion years for the amount of oxygen that we have today to begin to accumulate in the atmosphere. So my point is this: when photosynthesis evolved, there was not a lot of oxygen around. And you might say, well, what is the problem? Here is the problem. Rubisco would rather fix oxygen than CO2 in the Calvin cycle. In other words, if you have Rubisco, which remember that's the enzyme that is going to fix CO2 to RUBP, if oxygen is around, it will grab oxygen and try to put that on the RUBP. And that is a wasteful process. You don't get any sugar out of it. And so photorespiration consumes oxygen and it wastes the process, it wastes the resources of the plant, and it doesn't produce any ATP and it doesn't produce any sugar. So photorespiration is a bad problem. So here's what I was talking about. When you have plenty of carbon dioxide and low oxygen, then Rubisco will take CO2 and fix it the way it's supposed to in the Calvin cycle. But if you have too much oxygen, and remember this would occur if you had to close your stomata so the oxygen can't be released. If you have too high of oxygen because you're trying to preserve water from being evaporated, then photorespiration can occur, and Rubisco will grab oxygen instead of CO2, and that is not going to give you any sugar. So, you might say, well, I really don't think this is a big deal. What's the problem? Well, in many plants, photorespiration is so much of a problem that it can drain as much as 50% of the carbon fixed by the Calvin cycle. And on hot, dry days, that is a problem because obviously plants, it's a problem for the plants, but it's also a problem for people because we have 9 billion people to feed. So think about that. So let's go back to the stomata specifically, and let's try to figure out how they open and shut. So the stomata is actually the hole or the pore, and the cells that control the opening to that are called guard cells. And what happens is that these guard cells can pump ions into these cells. And of course, you know that water follows solute. And so the water's going to move in. And these cells, these guard cells, become turgid or swollen. And that kind of opens up the stomata. And so when they are flaccid, they kind of go back together and that shuts it. So that's the mechanism that opens and shuts the stomata. And I want you to look at the anatomy of what's going on here. So here's my stomata, right? So O2 and oxygen released in the plant, CO2 goes in. And then these cells, specifically the mesophyll cells, are going to be the cells where photosynthesis is occurring. All right, so we have these mesophyll palisade cells and these spongy mesophyll, and those are going to be where the photosynthesis is happening. So the xylem, remember, are tubes that carry water. The phloem are tubes that carry sugar. And you can see the sugar is going to be transported once it's made into those tubes. So let's talk a little bit about C4 plants. There are two other main types of plants that do a little bit different version of photosynthesis, and one of these is called C4. It's called C4 because CO2 is fixed onto a 4-carbon compound. And so that's where the 4C or C4 comes from. And what happens is that there is another enzyme, not rubisco, but this one is called Pep Carboxylase. And this Pep Carboxylase has a higher affinity for CO2 than rubisco does. In other words, it can grab CO2 even better than rubisco does. And so even when CO concentrations are low and oxygen concentrations are high, it will not do photorespiration. And so that is going to be a good thing. So C4 plants would be like sugarcane and corn and a lot of the grasses. So these 4-carbon compounds then, that is what happens when Pep carboxylate fixes CO2, they are stored into these special cells called bundle sheath cells, and then they are released into the regular mesophyll cells to complete the Calvin cycle. And so there is a very special anatomy that comes from this functionality that's called Cran's anatomy. So let's take a look at C3, right? So C3 plants are the regular photosynthesis. And I see my mesophyll cells, and I see I do have bundle sheath cells, and there's my vascular tissue. But in C4 plants, there's a very special relationship. So what happens is that my bundle sheath cells and my mesophyll cells are spatially very close together. And we're going to see that what happens is there's going to be something special that happens in the bundle sheath cells that is then connected very closely to a mesophyll cell. And these two cells are going to help with the process of photorespiration, as we'll see in a minute. So here's what happens. CO2 comes in C4 cells, and it is fixed via Pep carboxylase into a 4-carbon compound called all-oxoacetate. You may remember this from cellular respiration. Same compound. And so then this is transferred very quickly to a 4-carbon compound called malate. Malate is transported via the plasmodesmatas. Remember, those are kind of the openings that allow the cells to kind of shunt things back and forth. And CO2 is going to be released here, right here. And I've got pyruvate, which you remember also from cellular respiration. And the pyruvate, we're going to spend a little bit of ATP, and the pyruvate can be regenerated to go right back to get some more PEP. So CO2 in, Pep carboxylate is fixed there to a 4-carbon molecule. I've got malate here, and malate is basically storing the CO2 just for a little bit until we can get into the bundle sheath cell. CO2 is released here, and then it goes right into the Calvin cycle, just like a normal Calvin cycle, and makes some sugar, and the sugar goes right into the vascular tissue. So that is a way to circumvent photorespiration. So again, we see here, if you wonder what PEP stands for, it's phosphoenolpyruvate, all right, is PEP. And Pep carboxyl is phosphoenolpyruvate carboxylase. So this is just showing you what is happening. But remember, the main thing is that we get CO2 fixed right here, and then there is the CO2 that I can see here that I pulled in from the outside. It's on the malate. The malate gets transferred to the bundle sheath cell. CO2 comes right off. The malate was just kind of holding it for a while, goes right into the Calvin cycle. And you can see that I'm going to create some more NADP. I've got my pyruvate that I have to spend some ATPs. But remember, I don't have the waste of photorespiration. There is another strategy that the plant can use in even hotter, drier environments than C4 plants that are called CAM plants or CAM photosynthesis. And this comes from crassulacean acid metabolism. And the Crassulaceae is a family of cactus-like plants, and so that's where it was first seen, and so that's where we call it CAM. Now, what CAM plants do to prevent photorespiration and to keep their stomata closed and not let water evaporate, what they do is they don't have a special anatomy like C4 plants, but they do it temporally. That means time. So they keep their stomata open at night. When it's cooler, they open up their stomatas and all the CO2 gets in. They can store it temporarily, and I'll show you how they do that in a minute. Then they close their stomata during the day so water won't evaporate and oxygen can build up, because remember, it's going to use PEP, and CO2 is released from those organic acids and used in the Calvin cycle. So here's what's going to happen here. At night, this is a different strategy than C4, but the same basic output. CO2 comes in. I've got Pep carboxylase that fixes PEP to CO2. I get a 4-carbon acid that's temporarily there, and we saw those before, alkoxyl acetate and malate acid, but this time the malate or the malic acid is stored in a big vacuole. Okay, so it's like this big storage tank. And remember, that's really where the CO2 is. So that during the day, that carbon dioxide that was stored there, it can decarboxylate. That means the CO2 comes off, and there it's ready to go right into the Calvin cycle, and I keep my stomatas closed the whole time. So that works out greatly. And then this C3 acid that's left can be regenerated to go back into PEP. So again, this is just another picture. CO2 comes in, Pep carboxylates fixes. I get eventually a 4-carbon sugar called malic acid. It's stored in the vacuole, sits there during the night. The plant stores up a whole bunch of that. And then during the day, stomata closes, and I have plenty of CO2. And it doesn't matter that oxygen builds up because I'm using Pep carboxylase, and it goes straight into the regular Calvin cycle, and the normal C3 stuff happens. So this is just 2 different strategies that you see here. I've got the C4 plants that separate spatially in these mesophyll cells and bundle sheath cells with the special Krantz anatomy to circumvent the problem of photorespiration. And in CAM photosynthesis, I'm using temporal separation. So I get the CO2 at night, and then I store it up, and then I can get that CO2 back off and use it in the Calvin cycle. So this might be a good little review for you of all 3 different types of photosynthesis. Remember, the great majority of plants on Earth use C3. But think about climate change, and think about the effects of warmer climates, sometimes drier climates, and you can very clearly see that under certain conditions that may have been very favorable for C3 plants, for example, may not be favorable for those anymore in a warmer environment. And so you can see that climate change is going to affect things like what kinds of plants grow, and that affects people because plants are what we eat primarily as food, or we're eating animals that eat the plants that we grow for them. So that is an important thing to consider going forward. So don't forget that the sugar that is made in photosynthesis is not just sugar, right? So the plant can also use the raw products that it makes to make proteins and fats as well, and it can store the sugar as starches in structures like roots and tubers and seeds and fruits. And of course, in addition to food production, the oxygen that is a waste product goes into our atmosphere that we heterotrophs need to use. Thank you, plants, for your photosynthesis. So thank you, and I hope that's been helpful. And you may need to watch the video again, but hopefully that has been helpful. Thank you.